Hey guys, it's Friday for you guys. Today we are gonna be going over mitosis. If you already have biology, perfect. Then you should already have like a pretty good idea. This should hopefully just be review. If you're looking at the PowerPoint, we're gonna be going through slides 81 through 95. And y'all's assignment for the day is going to be to watch a YouTube video. I've got some questions put together, and then y'all are going to go through and answer the questions from the video. So pulling up the PowerPoint. Start out, good old meme. I love this. Basically just because I love this meme. But the first cell that underwent mitosis must have been like, and there was two of them. So we kind of went over this a little bit last class. In the last lecture, there's two different types of cells, like very, very basic. There's eukaryotic cells and there's prokaryotic cells. Prokaryotic cells were some of the very first cells that have ever been discovered. They're some of the oldest cells, and they're single-celled. They're very small, they're very tiny, they don't really move. Just very, very little tiny ancient cells. And there's eukaryotic, which are further formed. They've got all the different organelles that prokaryotic cells do not. And eukaryotic cells make up animal and plant cells. So, okay, perfect. Already kind of went over this, but just keep it going. Prokaryotic cells, super simple cells, very old cells. They have few internal structures, meaning like the Golgi body and the lysosome and the endoplasmic reticulum. Prokaryotic cells don't necessarily have all of those. And where do we find prokaryotic cells? Typically in single-celled organisms like bacteria. Those are going to be prokaryotic cells, and that's going to be just about the only place that we're really going to be able to find them. Eukaryotic cells are what we learned about last class. They have all of those different organelles, the endoplasmic reticulum, the Golgi body, the lysosomes, the nucleus, the nucleolus. I'm blanking on all the rest of them, but all those different organelles that we went over last class. Most living organisms are going to be made up of eukaryotic cells because the only other option is going to be prokaryotic cells, and those are little tiny single-celled organisms. So examples of eukaryotic cells: plants are made up of eukaryotic cells, animals, animal cells, and humans, which could technically go in the animal cells category. So today, well, I'll back up. This unit, we're going to be going over mitosis and meiosis. Mitosis, meiosis. Today, though, we're just going to stick with mitosis because, as you can see from the diagram, it's much simpler than meiosis. So we're going to start out with the simple, and we're going to go for a little bit more complicated next class. What is mitosis if you've never taken biology? Mitosis is a type of cell division. So cells divide, one cell ends up turning into two, as we can see from this diagram down here. And it ends up increasing the total number of cells. So rather than having two small cells, you end up with two of the exact same cells. They're completely identical, and they come from one cell. That's why they're completely identical. You're not going to get something different when you only have the same thing. It's like printing off a sheet of paper. If you print that sheet of paper twice, it's still going to be the exact same sheet of paper. Mitosis is seen for animal growth. So literally when an animal is growing in size. Its skin has to grow to be able to hold all the muscles. Its organs have to grow to continue digesting the food that it's eating and doing all the other organ jobs. Cell repair consists of mitosis. So like if you get a scratch and you slowly watch over the course of like a week or two, that scratch goes from being really big and kind of open and gross and red to slowly healing over. to the point that, like, at most you might have a scar. That's mitosis. That's the cells splitting apart and regenerating to fix something. Mitosis increases the total number of cells, results in animal growth, and the chromosome pairs are duplicated. So this means it's just like I said, you print out one sheet of paper twice, you get 2 sheets of the exact same paper. From the original cell, you get 2 duplicates. They look exactly the same. They do exactly the same thing. Their DNA is exactly the same. In mitosis, there's 4 different divisions or stages. The first stage is prophase. So this right here, this is seen as prophase. We're going from this to this. In prophase, the nucleolus disappears. Think back to last class, what does the nucleolus do? The nucleolus holds all of that DNA, and all that DNA is just kind of floating around, doing its own little thing. When the nucleolus disappears, the centrioles, which are the little star-shaped organelles, they move to opposite ends and fibers start forming. Also in prophase, the chromosomes, they align to form the chromosome shape that we always think of. It's no longer this tangled mess of DNA. It's these little crisscrossed X pairs. Then in metaphase, middle. So in metaphase, the chromosomes align themselves in the middle of the cell. You can start seeing the spindle fibers forming. So right here, we have the centrioles, and these spindle fibers start forming. The spindle fibers are attaching to the different areas of the chromosomes. So here's 2 different diagrams of prophase. Here's like a computer generated kind of diagram, and here's like literally what prophase looks like under the microscope. So in prophase, the nucleolus is going to start disappearing, dissolving. The centrioles are going to start moving to opposite ends of the cell, and you start seeing the spindle fibers form. Right here, you can see the DNA is kind of like twisting all around, doing its own thing. It's going to be getting into that little X shape. In metaphase, remember meta means middle. So in metaphase, the chromosomes line up in the middle. The centrioles send out the spindle fibers, and they attach themselves to the chromosomes. So here in the microscope, you can see everything is kind of lining up in the middle. It's not as perfect as it is in this diagram, but this diagram gives you the idea. Everything lines up in the middle, or as close as we can get to it. The third stage is anaphase. Anaphase is where the chromosome pairs start to separate. They're pulled apart. So we see we've got a green or a blue one here and a green one there. And then you've got its match right over here. These are exactly the same. They're linked up here, but they're exactly the same. So when they're pulled apart in anaphase, you've still got exactly the same DNA going on here. You've just got a little bit less of it. So in anaphase, they start moving away from each other. They're pulled apart. And then in telophase, they're just about separated, and the nuclear membrane starts to form right back around the new cells. Then the cell membrane is going to start to contract and break them off into 2 separate pieces. So the nuclear membrane is the membrane that surrounds the nucleus. What does the nucleus hold? The nucleus holds the DNA. So the nucleolus and that membrane disappeared in prophase, remember? In telophase, that nuclear membrane comes back. It reappears, and it holds all that DNA right where it should be. And then the cell membrane, which is the outer ring of the entire cell, starts to contract. So you can kind of see it right here. When it contracts, it ends up cutting it basically off from the other half so that you end up getting these two separate cells. So here in Anaphase, we can see everything is being pulled apart. The chromosomes have split from their little identical pair and they're moving to opposite ends with their own little centrosome. And here it is in the microscope image. Everything's kind of being pulled apart. And then in telophase, everything starts kind of being pulled apart. The nucleolus starts to form back around so that all the DNA gets back in the middle where it should be. And the cell membrane starts forming. And the cell membrane, you've got them forming on two different sides. So the further they form, they end up breaking off from each other so that they can pull apart and they can be their own two individual separate cells. And this is what it would look like in the microscope. Here's another diagram. The way I always remember prophase, or at least the stages for prophase, is it an anagram? I don't really know what it's called. I don't think an anagram is right. But I just can't remember what it's called. PMAT. I always think of it as PMAT. P, prophase, M, metaphase, A, anaphase, T, telophase. PMAT. So again, just to go through what happens in prophase, the nuclear membrane starts to break down and dissolve. You can see it kind of breaking down right here. It dissolves and the DNA forms in the classic little X shapes that we always think of. Then in metaphase, the spindle fibers start to attach to the DNA. Once they attach and they're in the middle, then anaphase will pull them apart. Anaphase will split the DNA sections up and pull half to one side and then half to the other side. Telophase, they're working on breaking up. Everything is where it needs to be. The nuclear membrane is starting to come back and the cell membrane is starting to form so that once the cell membrane fully forms, it's going to split these two cells into two so that we end up seeing this. So y'all's assignment, well, this is what we would do if we were in school. It's a lot of fun. You get some sprinkles and you'd have to show me what happens in the different stages. This is an actual picture from what one of the students last year did. We didn't really go over interphase or cytokinesis, so really quickly I'll go over that because you are going to see it in your video assignment right here. In interphase is the longest phase. You're going to get some time amounts in the video that you watch. Basically, interphase is like 3/4 of the entire mitosis cycle. Interphase is basically the cell is prepping itself to be able to go through prophase, metaphase, anaphase, and telophase. Interphase is just prepping. And then cytokinesis is what happens when telophase ends. So once it ends and it's trying to split up, it's not completely fully formed right here. So in cytokinesis, everything is fully split up the way that it should be, and it's forming the rest of those organelles that it should be forming. So the assignment is going to be posted on Edmodo. Here is the link. You're going to go through the video. I think it's like 10-ish minutes, so it's not a super long video. And it's 15 questions. The questions are in order. So if you miss number 1, but you get the answer to number 2, you need to rewind because you should get answer number 1 before you get answer number 2. Go through, work on that, and you guys have a good weekend. Stay safe, keep up all the good work, and I'll see you guys soon. Bye, y'all.